Hello, my name is Rachael Richardson. I'm a Principal Research Fellow at the Bionics Institute. I'd like to talk about some of the work that I'm doing at the Bionics Institute, where we're aiming to improve the precision of bionic devices using light. And I'm looking at this research from the aspect of hearing loss. So a disabling hearing impairment is defined as a loss of greater than 40 decibels. And currently there are 466 million people living with this kind of disabling hearing impairment. And this is projected to increase to 900 million people by the year 2050. And this kind of disabling hearing impairment is the type that actually affects your everyday life. And it actually has a bigger impact on people's lives than we can imagine. It affects social interactions, such as with your family, your friends or your colleagues. And this can have ramifications across all aspects of life, including your job prospects and education. So it's something that we'd really like to address. Now, currently, treatments for hearing loss are rather limited. So if you have a mild or moderate hearing loss, you might be fitted with a hearing aid, which amplifies the sound so that you can hear it a little bit better. If you have a severe or profound hearing loss, you might be recommended to be fitted with a cochlear implant. Now, cochlear implant, sort of the workhorse of the device is down this end. It's really, really tiny. These 22 electrodes or up to 22 electrodes are implanted into the cochlea. And these electrodes provide electrical stimulation directly to the auditory neurons. This means that the hair cells that would normally enable you to hear can be completely bypassed, because if you have hearing loss, they're probably damaged or lost. So we can directly activate the spiral ganglion neurons of the cochlea with these electrical stimuli. Now, there's a lot of people who use the cochlear implant, over 500,000 people now worldwide, and this enables people to understand open speech. That's just like this kind of speech, your everyday talking with people, sentences. And the way it works is that the sound input is processed into discrete frequency bands, and these are mapped to one of the electrodes along the array. And they provide information about the pitch of the sound, either by the place where it stimulates, so whether it's electrode 3 or electrode 10, and also the timing of the electrical pulses. So timing, the rate can also provide information about the sound. The thing is that with cochlear implant, there's actually quite poor speech comprehension when there's background noise. And this is the kind of noise you might experience in a restaurant, for example, where there's a lot of babble of talking behind you, or even just everyday background noise, traffic, or all sorts of the environmental sounds that you might experience. And there's also this really limited ability to convey music via a cochlear implant. And this is easy to imagine in a way, that because you're trying to encode all that richness of sound via just 22 electrodes. And when you think about it, I mean, even a piano has 88 keys. So how are you going to sort of transmit that information with just 22 electrodes? And one of the main reasons for some of these limitations of cochlear implants is the spread of activation. So when you put the stimulating electrodes into the environment of the cochlea, you get current spread. And this leads to channel interactions. So for example, if you look at this electrode here, instead of just activating the nerves nearby, right in front of it, the current spreads and you get activation that's much more broad than the electrode itself. And this electrode here similarly has a broad spread of activation. And so it would be very hard to activate these two electrodes together, because this population in the middle will be receiving information from each electrode. And this is where that fine temporal structure of the acoustic stimulus can become blurred, because this neuron here, it will be very hard to give it some really fine timing information from this electrode when it might also be getting it from this electrode. And you can kind of see how that's going to really blur the signal. And so currently this limitation is overcome by stimulating each electrode sequentially, so they're never activated at the same time. But this means that we lose a lot of the fine temporal structure of the acoustic stimulus. So one way that researchers are attempting to overcome this problem is to use light to stimulate the nerves. And light can be focused, and it's not subject to the same current spread as an electrical stimulus. And so there's the possibility that we can get much more precise activation of the neural tissue. Now, if you look at infrared neural stimulation, so you put infrared light into the cochlea, and what the researchers found was that there was this very precise spread of activation recorded in the brain of these research animals, and it actually looked very similar to an acoustic stimulus. So this was quite exciting when we're looking at spread of activation using a point source of infrared light. We're finding that we could get much more precise neural activation. And one of the positives about this technique is that you don't have to do anything to the neurons, you don't have to modify them in any way, they just respond to the infrared light. However, it uses really high power to get this effect. And this can lead to tissue heating, which obviously might be damaging to the neurons. And its actual mechanism of action is a little unclear. And so in response to this, some researchers, including ourselves, are turning to optogenetics. Now, this is a technique where genes for light-sensitive ion channels called opsins are introduced into specific nerve cells so that we can control their activity precisely using low-power visible light now. So it's not infrared, it's in that visible light range, and in fact, blue, for example, is a common colour that's used to activate these ion channels. So in this example here, the opsin is called channel rhodopsin 2. This is one of the most well-studied of all the opsins. And when blue light comes in contact with the ion channel, a conformational change occurs and the ion channel opens. It allows ions to flood into the cell, which can initiate an action potential in that neuron. And now, when researchers used channel rhodopsin 2 in some experimental animals and then came along with blue light to activate the nerves, they found that they got very precise activation in the brain. So this is a kind of a spatial tuning curve, and it's very sharp, and it looks like the acoustic stimulus. This is just normal sound. This is in contrast to electrical stimulation, which just sort of lit up a really big region of the brain there, so you can see that all that spread of activation. So this technique seemed to have a lot of promise, but I guess its main downfall is that it does require a genetic modification of the neurons to start with. But overcoming that, it does use very low power, and it has a couple of other advantages, and one of these is cell specificity. So in a way, because we are using a genetic modification, that means that we can decide, well, maybe we just want this population of neurons to express it and not this one. So we can use promoters to be very specific about what cells express these opsins. And so in this little diagram here, you can see that with electrical stimulation, all the neurons that are in contact with the electrical current are going to be activated. So there's really no way to get any specificity here. But with optogenetic excitation, you've got these two nerves here. This nerve expresses the opsins and this one doesn't. And so when you come along with your blue light, only the nerve with the opsins is activated. And that's where we get that specificity. So you can imagine if you have a mixed population of neurons and you want to be very specific, you only want to activate one population of nerves, you can do that with optogenetics. And this is something you can't do with electrical stimulation. And another positive aspect of optogenetics is the possibility for inhibition. So there are some inhibitory opsins, quite a few, in fact. And for example, if you take a population of nerves that are normally firing, maybe in the case of, say, chronic pain or something like that. Now, if you introduce opsins into, again, a specific population of neurons, now when you come along with yellow light in this case, you can silence that population of nerves and with that same specificity. So those are a couple of the exciting things about optogenetics. Now, at the Bionics Institute, we're taking this research one step further, and we're proposing to combine the two techniques. So we're going to use an optical stimulus and an electrical stimulus together. And what we're hoping to do with this is to use some of those positive aspects of electrical stimulation, because there are a couple of positives about electrical stimulation. And one is that you can stimulate nerves with very high rates, and the nerves will faithfully follow those pulses that you apply with high speed. And the other is efficiency. Electrical stimulation doesn't use a lot of power. So we're hoping to exploit those aspects of electrical stimulation, but to combine it with that precision of the optical stimulation as well as that cell specificity. So in this diagram, you can see, with, as I've said before, with the electrical stimulation, you get this current spread. You get a lot of the nerves being activated. And with optical only stimulation, the evidence is suggesting that you get this very precise, discrete area being activated. And I've indicated here that we're using, say, a very full strength blue light here. But without our proposal to use hybrid or the combination of those two stimuli, the optical and the electrical together, we will be able to drop down the intensity of that light and also use reduced electrical current. And we're hoping that we're still going to get that precision and all the other good aspects. So to implement this research, we decided that we're going to need 4 aspects delivered initially. So the first is to have a reliable method to introduce an opsin gene into the neural tissue. We also need a device to deliver the light and electrical stimulation to the neural tissue. We need a hybrid stimulation strategy. And at the end of the day, we're going to have to actually demonstrate improved outcomes for neuromodulation. There has to be a benefit or something that's better, a better outcome than electrical stimulation, which is the current gold standard. So looking at that first aspect, which is introducing the opsin gene into the neural tissue, well, for lab-based research, we've got a couple of options, and one is to use transgenic mice. And this means that through their genetics and interbreeding particular mouse lines, we can enable opsin expression in the spiral ganglion neurons of the cochlea. So all of these nerves that are coloured green in this picture express the opsin. And if you're not familiar with looking at cochlear tissue, I'll explain that many of you would know that a cochlea is shaped in this spiral. And if we cut through that tissue, we get an image that looks like this, and you can almost imagine the spiral going in and out of the screen there. And so you can see a lot of repeating elements spiraling up through the cochlea, and we're looking at just one of these parts of the spiral here, and all of this down the middle here is the neural tissue. So this is what we're really trying to target here. And anyway, so you can see the spiral ganglion neurons here expressing the opsin. And so our transgenic mouse model, it's an ideal model just for having highly controlled experimental conditions, but clearly it's not something that can be used in the clinic. So we were quite keen to also have a more clinical model, which is gene therapy. And this is where we locally inject a viral vector into the cochlea, and which enables the expression of opsins. And you can use particular promoters on the gene transfer mechanism, and so you can get it expression in the exact tissue that you'd like. When we injected this into adult mice, we found we got some expression, but it wasn't. You can see some even individual nerve cells here. But we found by far and away when we injected into neonatal animals, we got the kind of expression that we were looking for. So it's a really robust expression in most of the nerves of the cochlea. So we're using this gene therapy model for studying more the clinical implementation of the technique. I wanted to point out at this point, actually, that maybe it sounds a little bit far-fetched, having to do a gene therapy. Well, in fact, channel rhodopsin is already in clinical trial for the visual system, and so optogenetics is actually being considered for clinical therapy. It's not as far-fetched as it actually seems initially. Then we need a device and an electrical stimulation. So currently in our mouse models, we're using a laser-coupled optical fibre. And I'm showing a bundle of optical fibres here. We're not actually using a bundle, we're actually using just an individual optical fibre, but it just looks all bunched together as a picture. And we're adding that, or we're combining that with electrical stimulation. So for the mouse, we have a 4-electrode platinum array, so you can see 4 rings here. And sometimes even for our research, we're just using a very basic single platinum wire to deliver the electrical stimulation. But as our research is progressing, we're looking towards making a multi-channel device that has the light sources interspersed with platinum electrodes. And so we're looking at using LED chips. These are really, really tiny. They're less than, say, 50 micron by 50 microns in size. So all of this onto about 2 or 3 millimeters. And so with some collaborators, we're looking at developing these LED chip arrays. You can see an array of 10 here, and they can all be individually addressed, so you can light up one at a time. And this is the array here, photographed alongside our mouse platinum array, and you can just to get that size comparison. So they're quite similar in size, and this, for example, is about 1 millimeter, so overall about 2 millimeters in length, which is perfect for a mouse cochlea, which is very, very tiny. And so with our device in hand, we have to look at stimulation strategies, because we've got 2 stimuli now to do. And so we're looking at optical pulses that are about 0.5 to 1 milliseconds in length. And often we're using sub-threshold levels, so that's a level that is lower than required to activate a nerve. And we're combining that with electrical pulses, which are typically these biphasic current pulses. And we've got a choice of where to put that current pulse. It can be at the start of the optical pulse. We can time it so that they end together. Or we can even time it so that the electrical pulse happens or occurs after the optical pulse. But the first thing I want to say about these optical pulses and the electrical pulses is that I've mentioned that we're using sub-threshold levels. So, for example, if we find the electrical threshold, as in this is how much we have to put in to activate the nerves, and then drop it down a little bit so it's sub-threshold. And if we do the same with the optical pulse, we obviously don't get any action potentials, but when we put the two together, that's when we get an action potential. So often, I just wanted to point out here that we're using sub-threshold levels of the individual stimuli to actually get the action potentials when they're put together. And so we had a look at the firing, the probability of firing when we do put the two together. So I'll just explain this graph. Here we've got the optical pulse. There's 1 millisecond optical pulse, so it's on and then it's off. And then we investigated various delays of when we should put the electrical pulse. So, for example, in here, the electrical delay is about 10 milliseconds after the start of the optical pulse. And we actually found that there was the highest probability of when the nerve was going to fire was when the delay was between, let's say, 10 and 20 milliseconds after the optical pulse. So it's more like this scenario where the electrical pulse occurs after the end of the optical pulse. And this has to do with the channel kinetics of opsins. So opsin ion channels, they each have their own sort of kinetics of opening and then closing. And the particular opsin that we're using in this research is actually quite slow. So it opens, it takes about 2 milliseconds to open, and then it actually takes about 18 milliseconds to close again. And during all that time, ions can be flooding into the cell. And so this means that during that 18 millisecond period, we're increasing the chance of that cell firing. And this enabled us to come up with our stimulation strategy. And that is that we'd like to use the optical pulse to raise the level of excitability of the neurons. So not necessarily to cause activation or firing, but we'd like to have the nerves primed and ready to fire so that when we do come in with an electrical stimulus, we don't have to come in with very much to those action potentials. And sort of a theory behind this means that because we're using less electrical current, we're not going to get as much spread of activation because we're just not using as much. So which comes to my next slide, which is outcomes for neuromodulation. So what are we seeing with spread of activation? So again, we take our animal model, and in the cochlea we apply the wool fibre and apply some light as well as some electrical stimulation. And at the same time, we've got a recording electrode placed in the part of the brain that responds to sound. And we've got a 32-channel recording array. And the key to note here is that this region of the cochlea that I've coloured in purple corresponds to this part of the brain, which I've also coloured purple. And this, the very top of the cochlea, the apex, which is coloured red here, corresponds to this more superficial part of the brain, which I've coloured red. So that organisation of the cochlea carries through to the brain, which means that when we're measuring from the brain, we can make inferences about where is being activated. And so these are the spatial tuning curves that we get for various different types of stimulation. So on the bottom axis here is the depth of our recording array, so over here. So as I said, deeper, it corresponds to the full region of the cochlea, and not so deep is the red regions of the cochlea. And along this axis, the y-axis is the intensity of the stimulus that we put in. So for an acoustic stimulus, for sound, we expect quite a sharp spatial tuning curve, and that is what we get, which means that even as we ramp up the sound, getting it louder and louder, it doesn't spread out too much. Whereas when we put in an electrical stimulus, it starts off a little bit sharp, it's not too bad, but as we increase that electrical current, it really, really starts to spread out. I'll skip hybrid for a minute. We'll go to the optical stimulation. So this is just optical on its own. Now this is an interesting case, actually. Now you can see it's a little bit sharp. It's also a little bit broad and it's a little bit funny in shape. You can see that, at least for the very maximum, the yellow sort of indicates that really the area that's maximally responsive to the stimulus. That's quite sharp. But there's actually a fair bit of spread. And we believe that that's due to the shape of the cochlea. You can, and I've tried to show it here, in fact, where in these regions you can see that, sure, it would probably be quite sharp, but the light could actually probably shine right through the tissue and start to activate these apical regions at the same time. And that's what we're seeing here. You can see this funny little blip here, and we're thinking, yeah, actually, the light's actually shining right through. And this is actually something a little bit sort of probably unique to the mouse cochlea. It's just so tiny that the light's just going quite through. We're not too concerned about that. But what I really want to talk about is the hybrid stimulation. So by putting the two together, which means we're dropping down the electrical current, we're not using so much light, we're actually getting quite a tight tuning curve. And that's shown in this graph here. So as I've mentioned, you've got this big jump in the spread of activation from acoustic to the electrical stimulus. With the optical, actually, we didn't find a whole lot of difference between the electrical and the optical, and I believe that's to do with the light kind of starting to shine right through the tissue or apical regions. With hybrid, it means we can drop down all of those levels and we get this tighter spatial tuning curves. Now, another aspect, another outcome for neural modulation that we'd like to look at is the temporal kinetics. And so I mentioned at the start with electrical stimulation that we can actually modulate the neurons at very high speeds. So we can apply electrical stimulation at very fast rates and all the nerves will follow along all of those pulses really well. But with light, with optical stimulation, particularly with optogenetics, and due to those channels opening and closing quite slowly, it means that they can't follow really high rates of stimulation. And this can be quite a problem for the auditory system, where we actually need to activate them quite at fast rates. And so we took some cells, individual cells, and gave them some pulse trains of optical stimulation and measured their responses. Now, in this example, it's not very high rates here. We're just using 20 hertz. And with optical light alone, no electrical stimulation, we get one response to the pulse train, and then after that, they didn't respond. But as we start applying more and more sub-threshold electrical stimulation, so not very much, just 30% of threshold, we can actually get those nerves following those pulse trains quite reliably. We found the same with the nerve population in the mouse as well. So this at the top was a single cell recording, and this is a recording from the neural populations. So in this graph, all of these dots represent nerves responding to the stimulus. And when the stimulus is very slow, just 10 pulses per second, or optical only, that's the blue dots, or hybrid stimulation, all of them, they follow along those 10 hertz really nicely. Same for the 20 hertz. All the nerves are firing each time we apply the pulses. Same with 40. But as we start to get to the higher rates, 80 pulses per second, you can see here that the blue dots, the optical stimulation, the nerves start firing at the beginning, but then they kind of drop off. They drop away, and they can't follow that pulse train anymore. But with our hybrid stimulation, they follow those pulses all the way through. Same with 160 pulses per second and 240 pulses per second. You can see the light stimulation starts and then it drops off. But the hybrid pulses are following all the way through. And that's summarized on this graph here, where this maximum following rate or entrainment or whatever you'd like to call it is much higher for the hybrid stimulation compared to light alone. And here we're not even using a particularly fast opsin. We're using one at the very middle of the range. It's not very fast, it's not very slow. So we believe we can apply this to even some of the faster opsins and get firing rates that are within that range that the auditory system needs. So just in conclusion then, I've shown you that using hybrid stimulation, we can decrease that spread of activation, get it more precise, and increase the maximum stimulation rate. And this is a potentially paradigm-shifting technology, so moving away from electrical modulation that we've been using for decades into something quite different. And what we're hoping is that it will lead to an increase in the number of independent stimulating channels for the cochlear implant. Being able to use two at once and have independent areas of the cochlea becoming activated. And this will enable us to deliver that fine structure of the fine temporal structure of the acoustic stimulus and hopefully just get a better outcome for recipients of cochlear implants. And so I've shown you that we're starting to address all those 4 points that we would like for our roadmap. One is a reliable method for the gene therapy, developing a device to deliver the two modes of stimulation, getting a stimulation strategy, such as using sub-threshold levels and how the timing works, and demonstrating improved outcomes for neuromodulation, which we've done so far for spread of activation and the stimulating rate. And as we take this into the future, we'd like to do some channel interaction measurements and see if this technique does actually lead to this increased number of independent channels, and also to take it to other systems where you also need precise neural stimulation, and one that immediately springs to mind would be the retina, of course. So I'd like to just acknowledge the team that have put this research together, who are based at the Bionics Institute, University of Melbourne, and Swinburne University of Technology, and all of the talented researchers who have contributed to the research, as well as our funding, which comes from Hearing Loss, the Garnet Pass and Rodney Williams Memorial Foundation, and the State Government of Victoria. So thank you so much for listening.